Hello, hello.
Hi.
Welcome to Microbiology Journal Club. I'm Fazlan. This is my colleague Danny Chan, and this week we're going to be talking about mouse model papers. We're going to be delving into two really interesting papers where we look at COVID-19 in mice. Um,
hmm Yeah, I mean, we've been covering in this reboot of our journal club, we've been covering a lot of COVID-19. It's very topical.
hmm,
We want to know the answers. We think other people might be interested in them as well. Yeah, and through this whole time, we've talked about monkeys, we've talked about hamsters, but we haven't talked about the mouse model, which is, the workhorse of scientific research.
yeah, I mean, it's interesting because there isn't a COVID-19 like mouse model that everyone's really settled on, and mice really play an important role in research because they are because you can't really, I mean, in terms of like for vaccines and stuff like that, the first step is in mice because mice are like they're small, they're easy to care for, they're cheap, and you can't model an immune system in a petri dish. So,
Mhm.
uh,
Well, I mean, people are moving towards that, right? Like, I think that's
oh,
the dream in some labs.
oh,
But yeah, right now, like all those different cell types, it's really hard to capture.
yeah,
Um, yeah, in some cell culture.
we just don't have that understanding of the immune system yet, and hopefully,
hmm
like at some point in the future, we will, and we'll be able to move away from mouse models. But at this point, we are kind of left with them because we still need to understand how things work. Let me see. I think
hmm
I wrote some stuff down. Yeah, and also like thing about mice is they're also genetically uniform, and we can alter their genetics. So there are lots of like different. So we've already learned quite a lot about how the immune system works from mice, and we will have all those models ready and available. And
Yeah. Or maybe, or sometimes more accurately said, I've heard people say we know a lot about the immune system of mice. Right?
yeah,
Yes, the general principles of the immune system as well, but also just like specifically mice, we know a
yeah,
lot.
yeah, and that's kind of I mean because also the mouse immune system is relatively similar to humans. So I've heard quotes around like kind of 80% similarity or something like that. But obviously
Hmm.
these numbers
Hmm.
vary depending on how you define things. But basically wherever there's usually a homologue for the immune system in the mouse system, or like something gene that does a similar function. So it's a good
hmm
starting point to understand like kind of the bare bones of like pathogenesis. So
Yeah. Yeah, and I think that's a really good point about like homologs, right? Like that's one of the ways in which people make the inference between a model and the human case, right? Is that like you're looking for something that maybe the protein looks the same or the gene sequence looks the same, it performs a similar function, right? Like those are the things that make it relatable. Because then you can say, okay, we saw this effect in the mouse, like that's likely what we might see in humans.
yeah, exactly. That's one of the like kind of amazing things about mice. That we can model quite a few interesting human diseases, but the problem with COVID-19 is that mice tend to be quite resistant to it. So
hmm
so
Yeah, we know. I think we've talked about before. They have a different like they don't have the ACE2 receptor, or at least the mouse ACE2 is not doesn't bind
mm,
to the SARS-CoV-2 receptor binding domain.
yeah, so that first step that the SARS needs to cause an infection just isn't there in mice. Which means that if we
hmm
want to
hmm
test out a vaccine, everything will look like a great drug candidate in mice because they're already resistant. So
Well, yeah, it's kind of. I guess that's an interesting. I was gonna say when you think about developing a mouse model, right? Like, really, it means I want to. Scientists are trying to make the mouse sick,
yeah,
right? And they want to make the mouse sick in a way that resembles human beings, because then they can say, okay, so now we can test things. Because if it doesn't make if we give this thing and the mouse doesn't get sick, then we can say that we avoided, right? Like what we think is human-like disease in the mouse.
yeah, yeah. So and once we have that human-like disease, we can basically try and understand what's happening. Uh,
hmm
and
hmm
so the two papers we're going to be looking at today are going to be both like approaching this problem of how to make a mouse model that represents the human condition, and they take two
hmm
quite different strategies. Um,
Yes. Yes. Well, and they're both focusing though on that spike protein and receptor binding
yeah,
domain, right? And the receptor on the mouse. That it's that area, but yeah, there's many ways to skin the cat, so to say.
yeah. So it's all about that relationship of like the SARS-CoV-2 binding to that ACE2 on the surface of
hmm
cells. So the first paper we're going to be looking at is going to look at at developing a version of the SARS virus which can attach to the ACE2 of mice. So the mice have their own different ACE2. SARS has an ACE2 has the spike protein that's designed to attach to humans. But what if we evolved the virus to attack mice? Which
Yeah, so this is interesting because last week when we were choosing these papers, I remember you said, "Oh, this one got published," but actually the one that we found as a preprint, it was like they specifically modified the virus, and in this one they're doing an evolution or like a passaging, right? Yeah,
which is
adapting.
Yeah, that's interesting. That's almost like modeling what happened in the virus before it spread to humans. So because
yeah, forcing the scenario, right? Like,
yeah,
those poor mice, they were forced into the wet market.
I mean, yeah, this is almost like, I mean, the thing I think said, because there's been a couple of studies like this that have tried to do the same thing, and they're almost all, I mean, if we did the other way around, it'd be bioterrorism, but
well, actually, this is funny because that's like there that is like a subject of some debate in the scientific community, right? When they did it with the flu and they adapted them into ferrets, right? And people were saying, "Oh, that's not a good thing to do because we're allowing like this virulent strain of virus to switch." But a lot of the discussion around that was that there just has to be a good scientific reason, right? Like I think that like to do it just for shits and giggles, right? Like is like that's not what you want to do. But if you're going to learn something, and especially in this case, we're learning and setting up a system that could help us fight an infectious disease, then there's some rationale to go down that path.
Yeah, exactly. And I think another concern was like this being, if someone could read your paper, then take those strategies to try and create their own version of the virus. That was another, the dual use concerns as they're called.
Right.
Yeah.
But there's like that's like I mean, maybe I don't want to go down the tangent too far, but like that's a really interesting topic, right? Because dual use is like, again, somehow like the use that you're saying, it could also be used for terrible purposes, but that's almost always the case, right? Like there's a lot of research can fall under that banner, and so I mean, from what I've seen now, it actually is quite a political like topic, right? To talk about dual use, because like who gets to say, right? Like what's the research that is going to leak some dangerous weapon secrets out? Because the scientists, for the most part, like I mean, we get some training, right, to say like think about this aspect. And so when people are choosing to go on these experimental paths, there are usually good scientific reasons. So then that line between what is unacceptable and acceptable is a bit murky.
Yeah, and if there's a genius reading your paper on the other end, it doesn't matter what they who knows what's going to inspire them. So you can't really say whose responsibility it is. So
exactly. But yeah, let's dive into the paper.
Yeah. Adaptation of SARS-CoV-2 in BALB/c mice for testing vaccine efficacy. So, this...
And BALB/c just being one of those, I think we said this last time, it's like a really old lineage of mice.
Yeah.
Very inbred. They know a bunch of different things about it.
But yeah, so the great thing about using inbred mice, I mean, is that they react to things in the same way. So they're almost like clonal. So the most of the variation that we'll be seeing in these experiments will be due to the virus or something like that, or to various other environmental factors to the mice rather than their own genetics. So that's the idea behind it. So I guess like the first like figure we're going to look into is like their passaging strategy. So how they actually turn SARS-CoV-2 into a mouse-relevant virus, which I think they can call MASC-P4, which is mouse-associated SARS coronavirus, I believe, or
yes, MASC-P6.
MASC-P6, sorry, yeah.
I mean, what very arbitrary. I think the P is probably the sixth passage.
Yeah, that's right.
That's what they're saying.
So yeah, they get some mice and they infect them with SARS coronavirus, and then they take out their lungs, mash them up, centrifuge them, take off the liquid that contains the virus, and then give them to other mice. Yeah, it's
yum.
Yeah, it's not an ideal situation for anybody, but yeah, and they did that like 6 times, and so in the second figure they give the viral loads of each like kind of passage. So essentially what these show is like a basic rough estimate of the numbers of virus within the lungs at each time. And like so between like each passage, you suddenly see there's an increase in the amount of virus that is present in the lungs until it levels up around like 10 to the 11.
Yeah. Which, I mean, that could be that in those from every passage, it would seem then, right, that the population of viruses gets a bit better about infecting the mouse
Yeah.
until it sort of bottoms out, and it's like, okay, this is as much virus that can sort of grow realistically in the scenario.
Yeah, they reach their pop cap and they can't like do any more damage. So there's a limit to how much. So they kept going until passage 6. So they at the end of that they had their mouse adapted virus. And I think like in figure S1A they confirmed that with figure S1A they have like these pictures of viral plates that show off like the titers. So that was just the viral loads is the RNA, viral titers is testing against live cells. And yes.
Yeah, this is we I was gonna say we talked about this. This is the plaque assays,
yes,
right, that they're showing in the supplement. And like this is the classic, right? For bacteria, we do plate assays where we look at colony forming units, and for viruses, we do this plaque assay, which is like basically an opposite of a colony, where a colony is a single organism that grew up into a spot. This is a single virus that killed a zone.
So yeah, we have these
cells,
plate of lovely cells minding their own business, and then suddenly a virus goes. So you dilute your virus down. So the ideal is you dilute to a point where you only get a single virus. And then that virus will like kind of infect one cell, pop it, and then infect some other. So each dot is supposed to represent one virus if you dilute it down enough. If you haven't, then you just get this, everything dies. So you have to like dilute it until you get to this plaque forming unit.
Yeah, and you can kind of see that, right? In the 10 to the minus 1, it's hard to count single dots there.
Yeah.
Maybe 10 to the minus 2 you can count a bit better. They probably counted from 10 to the minus 3.
Yeah, that'd be because like you always have to find the sweet spot when counting things, because the temptation for when I'm running these experiments is like to count at the lowest one, so I have to count like 3 colonies, but you actually want to have that kind of, you need to have like at least 30 colonies or something to get that kind of in the right range. If you're being obsessive about numbers, I mean, sometimes you can just look at the rate, the kind of factor. So saying 10 to minus 4 means that there's about 10,000 viruses in these lung homogeneous samples.
Yeah, and I think I just want to remind the audience again that, you know, we always say that we want to see the titers, right? Like this is the gold standard of knowing if there's virus in something, because every dot corresponds to an active viral particle, right? That's in comparison when you're doing like the RNA sequencing or the RNA tests, right? That's just RNA.
Yeah.
We don't actually know if those viruses are whole, if they're able to infect. So this is the plaque assay that we've been talking about, and I don't think we've ever seen a picture of.
Yeah, so it's great to have a paper that explains some of the basics. And then they do a study of like age, so C and D. They look at aged biopsy mice and young biopsy mice and the amount of viral loads in various tissues. So they look at lung, trachea, feces, intestine, heart, liver, spleen, brain, kidney, and serum. So they try to look everywhere for this virus. So one of the key things. Sorry.
Oh, I'm just gonna say, and you can see that they're going down that path now of trying to figure out, can we make a relevant model, right, for human disease, because we know that it affects aged people more than it does young. So they're trying to see that, right? Like they'd like to see it. I don't know if they actually end up seeing it. None of this figure, at least.
Oh yeah, I mean it's difficult because I know that the actual like passaging was done in aged mice, so and so they're testing in young biopsy mice to see that there's like, I guess if there's a similar sort of thing happening as well, and also like immunity changes as things grow. I think by aged mice, these are mice. The older mice are like kind of 9 months old. Younger mice, I'm not sure about, like maybe a month, maybe perhaps. But yeah, that's an interesting. And when you look at dissemination, you're basically looking for a systemic disease. So looking at if something shows that affects you in the lungs and shows up in your heart, liver, and spleen, suddenly you know that it's gone everywhere, and it's really severe. So that's almost another measure of disease severity. So if you see like a lot of that going on, then that does indicate that, okay, there's something interesting going on here that could give us some answers to certain questions.
Yeah, and I think the other thing is that we also we have some idea of what this looks like in humans, so it's also to make the relation. And previously, yeah, because this is one of the more recent papers,
Mm.
and so there have been. I'm not sure if I read it in this introduction or just when I was looking at other introductory stuff, but when they did some of the mouse models, when they infect them, if they've like
Mm.
matched up the ACE2 receptors or whatever, they find a brain infection
Yeah.
that ends up being fatal. But like that, we don't have a lot of cases of that in humans.
I mean, yeah.
So,
I think because mice
uh,
might be a bit more vulnerable because they've got this really big olfactory bulb that has a lot of like nerves going into into like the nasal cavity.
yeah, that section.
Yeah,
Hmm.
I mean, when I was working with Streptococcus pyogenes, with I'd see quite a lot of. Well, I saw like a number of times where mice would get like infected in the brain, and there'd be like this big like kind of when I dissect them, this big lesion there, and
Mm, mm,
and you'd never see that in humans, but it's just like I
right.
think it's just a quirk of the model sometimes that there are these
Yeah.
sometimes differences that can be formed, and I think like in the hamster study there was a sense that like the SARS had like a tropism, it kind of liked to go towards nerve cells, so it
Hmm.
wouldn't.
Hmm. Oh yeah, that's right. They did some staining even of that nasal epithelium, and they saw a little bit of
So
that.
if there's a slight like kind of like instance of it going towards nerve cells, then in an animal model that could actually have. I mean, I'm speculating wildly here, but that could
Yeah,
have more of
for
an
sure.
effect. Um,
For sure. And I guess the major point of having this data shown to us is that we don't really want to study that cause of that dying
yeah,
animal, right? Like if they're getting some sort of brain disease that like we don't really see, it's not a good model anymore of human disease. Like we want that human disease to be like what humans get,
but on the other
which
hand,
we know is like very lung heavy.
it can tell you quite something quite interesting, like because humans get that olfactory symptoms where we lose our sense of smell. So
True.
So so knowing that it attacks the olfactory bulb of mice and that could be more severe, then that does tell us something about the human condition. It's not the same thing because animals are different. So we could.
Hmm. Hmm.
So it's a one of those what.
And I think we had touched on this point before. It's like, and it just depends on the endpoint you're using,
Yeah,
right? Like if you're using death as the endpoint, then that's not very detailed enough, right?
yeah,
Because like it died of something that wasn't really what humans die of.
yeah,
But if you're using if you're using a different outcome, then maybe it doesn't matter, and this is just the feature of disease in the mouse.
exactly. So
Hmm.
now going to figure. The SB. So this looks in so S1B where it's a picture of lungs stained with for like SARS-CoV-2 in aged versus young. I think the main takeaway from this one is that the aged mice seem to have more SARS-CoV-2 in their lung airways.
Hmm.
Uh,
Yeah, visually speaking.
yeah,
But it's present everywhere. And I guess maybe also that it's like on that epithelial layer. That's another thing that we can observe.
yeah,
That it spikes in day 3 and it goes down. But we also know that from looking at C. So this is why it's in the supplement.
yeah. It's a pretty picture, so we get to look at it anyway.
Yeah.
And then we've got a much more complicated picture, which I had to do a lot of research on. Fig 1D, which show
Oh,
where
yeah.
I had to look up a lot of like lung cells so this like figure uses stains for very different lung cells that are present
Hmm.
there. So things that I used to call these Clara cells, but then I realized, well, then I figured out that they changed the name because the guy so these cells secrete like kind of various proteins and stuff for defending the lung, and they named it after this guy called Max Clara, who was a Nazi, who who
Yeah, I read that as well.
yeah, I
on
know. He
Wikipedia.
discovered them using the tissues of executed victims of experiments. So this is why we call
Uh-huh.
them club cells rather than Clara cells because
Yep.
I think there's a movement to say, you know what, maybe we shouldn't give Nazis credit for this. Maybe we
Yeah.
shouldn't do that. Like,
Yeah. Yeah, I think that's really nice of the I think that's a really progressive action for the nomenclature people to go after.
yeah,
I like that when I read it.
yeah. I feel like it's a good thing when we say, you know what, we're not going to endorse this murder. If you murdered people to
Yeah.
get your data, we're not going to really
Yeah.
give you credit for it. which I think
And I mean, it's interesting too because for everyone who went through school and learned it, it's also like this moment where you're like, I read club cells, I'm like, I've never heard of that before, and I looked it up. I was like, oh, Claris's, and you like learn a little bit of that, and you're like, oh yeah, that history of science, and like
yeah.
everything is not everything is sugar, spice, and everything nice. Like there's a lot of stuff that is ethically confusing, right, and complicated to wade through.
Oh yeah. I mean,
but anyways, Claricels or club
club
cells,
cells, yeah.
as they're called now, secrete, or they're sort of in the upper airway, and they secrete surfactant and some other mucus that helps line those upper airway. It's a common location for infections, right? I think that in flu, they also have some tropism for club cells.
Yeah. So club cells, they're the ones that are in red, and then we've got like the beta 4 tubulin. So that's so tubulin is like a big sticky like. Oh, no, I didn't mean sticky protein. I mean a big protein that's like a stick.
Yes.
That is formed in cilia cells.
Stick-like.
Yeah. So they're formed in ciliated cells, which play a big role in like managing your mucus. And so when you cough, it's because your cilia are like kind of pulling up all this mucus and all the like kind of crap caught into it, and then it allows you to expel like kind of all sorts of like debris from the lungs. What else is there? There's alveolar type 2 cells, which also manufacture surfactant and So, I mean, for viewers who don't know what surfactant is, it's very interesting, but a very like kind of important part. It's like a liquid layer that forms like they're like bubbles, but they have a structural purpose. So without your surfactant, your lungs kind of like flop over, and they don't like kind of have the same structure.
I mean, with surfactant, that's like they break up surface tension too, right? So if you like water beads up, your lungs are like they're moist areas. You don't want all the water to be beaded. You want like a thin layer of liquid, right, all around all the different cells. So surfactants help facilitate that.
Yeah.
And I guess importantly, what they were saying in the paper is that the beta tubulin is on this one type of epithelial cell, and then the surfactant's made by another type.
Yeah.
And they see co-localization of the SARS-CoV-2 protein with the surfactant-making cells, but not the ciliated cells.
Yeah. And they also do other things with like alveolar type 1s, which are in purple, and they're everywhere because alveolar type 1s are like your basic alveolar cell. They do all the gas exchange stuff. Yeah. And DAPI staining, because we all love DAPI staining. That stains for DNA, so
it's the nucleus of all the cells.
Yeah.
But so what I pulled to understand this paper was I picked up like this Martins et al. Early release pathology and pathogenesis associated with coronavirus disease. Basically, they did this in humans, right? Like they did autopsies on humans, and they looked at the same staining, like where are these SARS-CoV-2 antigens showing up in relation to cell types, and it is in these club cells. It is in the SP or the type 2, right, alveolar and endothelial or epithelial. And so like this D is very promising from the standpoint of human disease because they're showing us that it's hitting these cells. These are the types of cells that we see in humans that get hit as well.
Yeah, and I think that's a really important finding for this paper because it allows us to so this basically kind of validates that way of studying how SARS-CoV-2 like infects and pathogen. So I think that there's something that I'm just going to keep in mind for like the rest of the things because I think this is a really good figure and it really illustrates that.
For sure. Yeah, I think it's like this is the whole thing. It's like they're trying to find the differences and the similarities between humans. I think that like there haven't been any reports of like getting it in the heart for humans. Like that seems strange from C.
I feel like there were some things on like vascular disease related to humans, but not necessarily so.
Oh yeah, endothelial. Yeah, there was definitely some endothelial effects and the clotting effects. But yeah, they see them in hearts here. So you're wondering like, is that the right thing? And maybe they could fix some of those things with dosing as well, right? We haven't seen like the dosing curve of giving different doses might have different effects. They don't really show us that information.
Yeah, I feel like for this first one, they're really trying to get a big infection rather than doing the nuance. They're just doing this big like characterization to show like, oh, this is like something that happens in mice. Uh, so the next two figures I'm going to be looking at are let's see, figure 2A. I think that looks at the inflammatory response. So it's so H&E staining. So we've got like a look at aged mice and young mice, like almost like together. But so again, you've got some nice like so H&E staining stains for like nuclear.
The nuclei, yeah,
Yep.
yeah.
And
That's coming up purple in these, and the sort of extracellular areas come up with the counter stain.
yeah.
Pink.
And basically, this shows all the inflammatory cells going towards a destination. So inflammation is a sign of like a severe infection. So when inflammatory cells go through, they use all these like kind of enzymes to break through the tissue to get to certain places, and that can really wreck like your lung structure. So
yeah. So they're showing a few things, right? In the mock, well, you can definitely see in the mock everything looks fairly structured, right?
Yeah.
There's like these bubbles, right? Like thin layers of cells. The ciliated surface is like thick and like lush and like unbroken. But then you have like the later days infection, and you're seeing disorganization in those layers. And then you're also seeing, and again, like you have to zoom in probably a little bit, but you're also seeing hopefully like those polynuclear infiltrates.
Yes.
Right. That's the other. We talked about that, right? Like immune cells often have some polynuclear looking phenotypes. And that's what we're seeing infiltrating. And again, this is something that we know from humans that's ours.
Yeah.
Right? That this is the infiltrate that's filling up your lungs, that's giving you the pneumonia, making it difficult to breathe. And these mice are experiencing something like that as well on the histological scale.
Yeah, and this graph is essentially getting experienced pathologists to give each of these a score, and then we take an average of that score to compare them. But I mean.
Yep.
Yeah,
and it looks bad.
It looks
3 days
bad.
post-infection, there's something more severe than in day 5. So like we're also understanding now the spike of, or the sort of the progress of the infection, right?
Yeah.
3 days later, it spikes and then it goes down.
Yeah, and again, you see similar in young mice. It somewhat looks. I mean, I'll be honest. I can't interpret these as I can't really interpret the difference of these, but yeah.
I trust the graph, right?
Yeah, I have to trust the graph.
Yeah, tell me what the experienced histologist said.
Yeah, they say it looks bad. I think I believe them.
It definitely looks bad. I mean, they don't compare between aged and young. They're just sort of looking aged as if it was a model, young as if it was a model.
Yeah.
Right? Which one could give us the disease that we want to see?
Yeah, they're showing them side by side basically, but without actually doing any direct like comparison. So.
And they also do like in then B and D. Then they're gonna look at a panel of cytokines that are also made, right? Inflammatory cytokines.
Yeah, that's right.
And that one's a little bit different, right? They see some differences there between young and old.
Oh,
at least from this. Yeah.
Yeah. So like so I'm looking at the ones with all these nice limits. So I mean, which one am I looking at? Because I think I might be looking at figure S. No, it's figure 2B is the one with. Oh right, no, I see. Because this paper jumps around very very weirdly. So yeah. 2B and 2 like D I've kind of put up together, where they've got this. I'll be honest, I don't like these graphs. But basically, because like firstly the scales are on like the age mice is a bit jacked up because they've got like blue at the bottom and then blue at the top. So and it's a bit hard for me to interpret these because again the colors, but fortunately in the supplementary figures they have them in actual graph form. So in supplementary S3 they show that data in the form of a graph, which makes it a lot clearer like what the differences are.
For sure.
So I mean I get the impression that they wanted to show these graphs, but for space reasons they must have like compressed it down to that other format.
Right. Yeah, no, I feel like it must be a mistake that blue is at the top of B, right?
Yeah.
It should be red up there.
Yeah, I feel that it definitely feels like that was like an error there, and yeah.
Well, we can like just double like I mean this is like the double check, I guess, right? Like you can look at B like what IL-5
I
in aged mice.
yeah, and
IL-5 in aged mice, 3 days post-infection versus 5 days post-infection. Yeah, like
yeah,
those are high.
yeah. And like yeah, doing like I mean, you can sort of like do a look and try and compare, but it's again like it's hard to look at an entire panel. I mean, they're mostly pro-inflammatory like cytokines, but they are doing slightly different complex things. So while there is this trend, there are going to be like small variations in that have
Yeah.
other effects. But I don't think they're not drilling down deep into it. They're just like giving.
No, but they're presenting it for people who are experts in immunology, right?
Mm,
Who might know more insights about that human disease. Maybe they could say something, right,
yeah,
about how these two models, how the old versus the young mice stack up in terms of their immune response.
yeah,
Mhm.
yeah. That's right. So
Yeah, but for our takeaway, inflammation happens.
yes. Yeah. So yeah, I mean, I looked at figure S2A was quite interesting to me as well because that looks at that like does dying of inflammatory cells as they cause an infection, and there is a lot,
Mm,
and it does look quite dramatic in the old mice at 3 days because like in the old mice there's this like big green like kind of blob
mhm.
of SARS-CoV-2 there that you don't necessarily see in the young mice, which is
Sure,
but then again, it is
sure.
just one image, so you can pick it and
Yeah,
choose.
that's
But
just an
yeah,
image. But I feel like the reason they're showing us this though is for that cleaved caspase,
yes,
right? Like they want to show us that there's apoptosis happening.
yeah. So cleaved caspase, that's up in red, and it's a sign that of apoptosis. So it's programmed cell death. So when
Hmm.
which is different.
And also I think that that's like apoptosis is like tightly linked as well with the whole inflammation. Like
Yeah,
this is crazy, right? Because inflammation is like this very complicated like bunch of immunological terms, right, in the scientific community, but it also gets slung around in popular
oh yeah,
popular like speech and parlance.
yeah,
And so, you know, like inflammation not always bad, right?
mm,
Like right? It also helps control disease.
yeah,
And often disease is some like crazy balancing act between the inflammatory response and the progress of the foreign antigen of the foreign, pathogen. So, anyways, I think just important here to say that it is causing some sort of apoptosis,
yeah,
inflammation style event, which we would expect because that's something that causes this huge immune infiltrate.
yeah. And they do like make an effort to like look at the cells like macrophages, so CD103 dendritic cells, CD163 are macrophages, and CD3 are T lymphocytes, and
Hmm.
and it is interesting to and also I like the pretty colors. Okay, I'm not gonna lie, that's
Oh, they're great.
yeah,
Yeah.
um, yeah. So
And so they do say that it's macrophages that are undergoing this cleave caspase 3, right?
I think so. Yeah. Yeah. I mean, it looks like that slightly from. I mean, it's difficult because like
Yeah, it's pretty small for us to look, but I did think that they said something
yeah, that would make sense. Because again, that's
self
something.
recovering. In addition, they did this massive cell death as evidenced by this cleaved and significant inflammatory cell infiltration. Oh no, they don't say anything about like co-localization. There's just a bunch of cell death and then there's a bunch of these immune cells in there.
Yeah.
And so actually then the CD103, the CD163 and CD3, that is basically in addition to the H&E, right?
Yeah.
So we could see the polynuclear infiltrate, show us the molecular markers that say those cells are in there, and we can see those as well, like confirmation of that.
Yeah. Um. So these past few things have basically to demonstrate that this shows a very like inflammatory kind of human-like disease. I'd say.
Yeah,
Yeah.
yeah, I think like they're trying to establish with these figures, all in all, that the mice get this disease, that it has a course, right, where they infect the tissue tropism looks similar to what we see in humans. So does the immune response.
Yeah.
Yeah, in these broad strokes.
I mean, they did try to look at weight loss in mice, and it's it's odd to interpret weight loss because I mean for young mice they tend to be gaining a lot of weight early on, so so percentage weight change. So I mean in some cases you can see a flat line of weight change and interpret that as saying okay, well in healthy mice it would have been, but then again they look at mock and infected and there isn't really much change in weight except in like the aged mice. But
yeah, this disease is not that severe, right?
Yeah.
Like there are all these responses, immune ones that look similar to the way humans would respond, but the mice themselves aren't super phased.
Yeah, they're still eating as much as usual. They seem fairly happy, but I mean,
Yeah.
So yeah, that's.
But I guess like the thing is, if we're using this as a model and we're gonna sack the mice anyways to look at this pathology, right? Seeing the pathology go down or up, like that's closer to the human. We don't have to see death as the endpoint, right? We don't have to see sick mice as the endpoint. We can just think about the mouse is just a immune petri dish, a fancy immunological petri dish, and to read it out, we have to sacrifice those mice.
Yeah. And so the next stage is where they look at the actual mutation that the SARS-CoV-2.
Yeah.
Yeah.
Why does it even happen? How did their weird passaging strategy work?
Yeah. And they find like a couple of mutations that are going on. So I'm looking at Figure 1A, and I've also put it mashed in Table S1 as well. Sorry, Figure 3A in Table S1.
Yes.
Yes. So that really drills down into the where the mutation was happening. So so the first thing is to like figure out what's changed and how that change the ability of SARS-CoV-2 to attach to a certain receptor.
Yeah. Yeah, so they had those pooled virus, right, lysates that they were instilling into mice over the different times.
Yeah.
And so then they just pulled out that same bunch of virus and they sequenced it to figure out what's going on. And they kept seeing this one. Oh, so non-synonymous changes, those are ones that don't affect amino acid sequence. Or sorry, synonymous changes are those that don't affect amino acid sequences, right?
Right.
And then it's the opposite. And then non-synonymous are ones that are affecting that in some way.
Yeah. And they found like one that was particularly in the receptor binding domain that they felt was quite important. And figure like 5B, I think it is, or no, 3B.
Yeah, 3B.
Yes. Yeah, where they like, so what figure 3B shows is like a diagram of the molecular structure of the receptor binding domain, and kind of how like the change in like one amino acid at the 501 position from I forgot what N stands for. I remember Y is tyrosine, but I
don't remember either.
Yeah, so the change from N to a tyrosine is apparently like does change the binding affinity by a fairly strong margin, so that change is thought to be the reason why this receptor binding domain can now bind to the mouse ACE2.
And I think it has come out in this figure, maybe they have in a supplement, but they also, when they go back and they here it is, it's like it's in D, right? They're actually sequencing back in all those passage ones, and they see that it's like the prevalence of the one that they are hypothesizing like leading to the greater infectivity, the proportion of that sequence is like rising.
It's quite, I mean, like Passage 2, it's like 50-50, and then suddenly you can see it like out-competing in like real time. And I mean, this is interesting from like just an evolutionary perspective of looking at how quickly like in this situation, this, because it's present in like the first, like after the first passage really, which is interesting, and that kind of like almost speaks to how adaptable this virus is.
Well, it's an RNA virus, right?
Yeah.
I think that's a feature of things that go through any RNA. So whether that's retroviruses or direct RNA viruses, right? That polymerase is thought to be have less fidelity. So it makes like some baseline errors, and so that can lead to favorable that can be a source of variation on which selection plays a role. And in this case, we used artificial selection to get a virus that's going to be useful for our purposes. But the idea is that in nature, it's adaptive for the virus.
Yeah.
to be able to go and in this case, right, maybe cross species barriers, right, as it does in the zoonotic case where we think it came into humans.
And the interesting thing is, like, keeping to back of mind that first like diagram of the number of viruses per passage, and like how the big jump kind of happened between like P2 and P3. So let me double. Yeah, there was like a massive jump in. So
Yep.
between P2 and
It's
3.
the same thing.
Yeah.
Yeah.
So there's this like kind of almost like
That's when that genotype takes over.
yeah.
When the genotype takes over.
Yeah, that inflection point in because I guess before then you have all these other viruses that trigger the immune system that causes the disease to be like kind of dealt with very quickly, but then suddenly you got this inflection point where you get more of enough of this virus to cause a disease, which I think is like kind of a really fascinating takeaway from this. Yeah. So C is interesting because it shows like kind of the tropism of SARS and ACE2. So the binding of it to ACE2. So they've got like some cells in red that are expressing ACE2 and cells in green showing SARS. So basically wherever there's yellow in this like overlay is where they're both together. So that's
yeah, and this is just another way of them getting is it binding?
Yeah.
Is this thing binding?
Yeah, and I think they show.
And it is.
Yep, it is.
Yeah, they show the co-localization.
Yeah, and let's see what. And then they test out their own pet vaccine candidate.
Yes. And I think this is why at the end of the day, right, like they're happy showing whatever they, for whatever reason the model may have issues, right? Like the limitations that we may learn from this model coming from the viral loads and the tissue cell specificity, they're pretty good, right? Maybe not exactly human, we don't know, right? But pretty close. But they're gonna tie it back, bring it home for us by saying, oh, but we can test something in this system, right? Because we see these markers of disease, it means we can also do an intervention and hopefully see those markers of disease go down.
Yeah, I mean, this is like the kind of value added that most model papers don't really get very, very far through publication. People go like, oh, you can create a model, but what does it do for me? And this vaccine is like, what will this model do for us? And this demonstrates, oh yeah, we can test vaccines using it.
So, which is, what I think this is cool from the perspective of they changed the spike.
Yeah.
Right? Like if they changed the spike so it interfaces differently, I guess the receptor binding domain, but that has no effect on the antibody that is produced by this particular vaccine strategy.
Yeah, this vaccine uses the human RBD, so it doesn't use their evolved one. This is a human vaccine they're testing in mice with this against this this relatively different coronavirus. So actually that can be very interesting to see whether that can induce the change. Well, that can still have the same effect.
Well, I mean, it's promising for me to see that because it means, okay, well, that it's not so specific, right? That the protective antibody is going to be specific for that receptor binding domain region. Right, there's a bunch of different receptor binding domain regions that get recognized and can be neutralized independent of the one making connections here. Yeah. So it's nice to see this because it is also helping us understand, because that would be one of my reservations about the model being like, well, they changed the the spike protein of the virus. Like how can we test
exactly
vaccines in the case where they changed it? Well, they didn't change it enough to make any difference, at least in this example.
Yeah, exactly. I mean, this is really important to demonstrate that exactly what you said. That this is still a relevant model.
Right.
So yeah, in figure I think 4B, they purify the protein vaccine. So this is a vaccine that's made out of human RBD attached to an inflammatory portion of an immune cytokine. It escapes me at the moment, but so they purify the protein and they kind of check out the amount that they check whether the receptor binding domain can still can attach to human ACE2, and they do kind of show that in Fig C.
Oh yeah, no, it's attached to FC, right? That's the that's the constant region of antibodies.
Yes, yes, that's right. Yeah, I completely spaced on that.
Yeah. So what
Yeah.
their receptor binding domain attached to FC?
So
It's odd. I mean, I guess it works.
yeah, I
I'm
mean,
just trying to imagine how that's supposed to work.
so that yeah, it's I guess it's because it'll like activate the immune cells, but yeah,
Yeah, it could be that immune cells grab onto RBD, intake it, and then present it on the outside.
yeah.
Right? So this could be for macrophages is my
Hmm.
there's like lots of there there's probably a better person to hypothesize on how it works, like who has a bit better knowledge of the immune system than I. But I would say, that's my thought. FC macrophages will grab FC because it's an
Yeah.
antibody, right? Constant chain. They're going to phagocytose it, right? They're going to process whatever's attached to it, saying that this is an antigen to respond to. That's going to get presented, and then we're going to get some immune response to that RBD.
Yeah. Uh, yeah, and then they show their results in like in Figure 4, so Figure 4A, where they test like kind of like the PBS and like one that had one vaccination and then a booster vaccination as well. It seems,
Mm-hmm.
and they look at like the Figure A is showing the IgG titer, so that's just an antibody that will attach to this particular antigen. Well,
Mm-hmm.
in this case, I think they just go straight for the spike protein, and they find that yeah, after a booster, there's an increase in IgG, which is good.
Yep.
And what we want to see is in B, so that neutralizing antibody titer. So
A functional way of looking at it. So now with actual virus,
yep,
does the antibodies coming off of this prevent those viruses from infecting? And they do.
yep, they do. And then they look at the viral load, which is the RNA copies, and they find that the RNA copies are low as well compared to
Hmm.
control. Um,
And that's C. That was in an animal then? C now? Yeah.
see,
So then that's in the lung,
yeah,
and they take up the lungs. Hmm.
yeah. So these are taking out the lungs and then mashing them up and then seeing what comes out, I guess. Um,
Yeah.
yeah, our profession is grim sometimes. Um,
Yeah.
and then
um,
figure D looks at it shows us a nice stain of like SARS-CoV-2 protein in like vaccinated versus non-vaccinated, and the green dots are in the non-vaccinated, and so the green dots represent SARS-CoV-2, and that seems like yeah, it shows that there's a difference.
hmm Yeah, there's less.
Yeah. And again, the final figure is like showing inflammation, which is what we really care about because we don't want the immune system going crazy. And it looks like the immune system
Hmm.
goes crazy in the PBS with all these like purple blotches indicating that there are these inflammatory cells, and the RBD, it seems that's fine.
Yeah. Yeah, so
Yeah.
this is a promising paper, right? Like this is a way of changing the virus to give us a disease, the
Yeah.
model of disease, right? And then they wrap it all up and they show us that it can give us some useful information. It can
Yeah.
test some
I
interventions.
mean, there is only one thing that I'm kind of. It's a nitpicky thing, but it's just
Sure.
that like they. I mean, the amount of times they compare the actual SARS-CoV-2 and the mouse adapted SARS-CoV-2 in this paper is zero. So,
Mm.
and I
Hmm.
would have liked to see that just for like kind of just for comparison to see like because again like that resistance
Yeah.
would tell us something about like the difference between like the. But I mean,
Well, like for example, they do mock it for like we're thinking about figure two,
yes,
right? Where they show us some of the disease,
yeah,
right? The immune infiltrated the disease. They show us mock, which means no infection.
yeah,
But what is it? What about unadapted?
yeah.
Right? Like, how do we know that this is not just like a general immune response that that mice make with whatever comes across, right?
Yeah,
That they see.
that's the thing. I really wanted to see that there, but they don't show it, and that kind of make what
Hmm.
they do is they do like reference that. Oh, look at this other paper, and I feel like you shouldn't use another paper for your control. Like that's like oh
Yeah.
yeah, if you want a control, just read someone else's paper. I'm like that's if you want a control, you need to keep all the conditions the same. So I would have been
Sure.
like oh
Their hands and their facility, right?
yeah, I mean, mouse strains can differ between different labs. I mean, even the same mouse strain can pay fairly differently depending on what university is hosting it sometimes. So it can be
Mm.
so, and also sometimes people in literature don't necessarily tell the truth, so it's nice to double check.
Yes. Yes.
So
Yeah, that definitely is something that Yeah, that you would hope is obvious, and that there weren't any of these effects inside of
Yeah,
the mouse that was infected with mock. Because again, I guess you could see like the titer's so much lower,
yeah,
but yeah, they never really show us what that looks like on the disease side.
yeah. So that's like kind of an unfortunate wrinkle, but it's I can see why that people would make that kind of but people would like make that cost cutting measure because they go, oh, it's like kind of thing that's accepted that the mice are
Yeah.
less susceptible. So I can understand why it was left out of the paper. It's just for my own edification, and for I'd have really liked to have seen that control. Um,
Hmm. Hmm.
yeah, and I guess that finishes off that paper, and that brings us to
Yeah, absolutely.
our second paper, which takes in a completely different strategy to this, where
Still the spike though, right?
yep,
Still
still
trying to change the interaction between the mouse, which doesn't normally interact with that spike protein, and yeah.
yeah, except on this side they're changing the spike protein of the mouse to make it more human. So
Yes.
we're looking at now a paper called a SARS-CoV-2 infection model in mice demonstrates protection by neutralizing antibodies. And I'm going to start off with a graphical abstract because I like graphical abstracts mostly. But yeah, so the basic structure of this paper, well, their model is they infect mice with an adenovirus that can make them express human ACE2,
Hmm.
and then after they're expressing human ACE2, they get infect them with SARS-CoV-2, and then they look at the disease progression from there.
So these mice are getting double virus.
Yeah,
First, the virus to make them more similar to us, give them the way for for the SARS-CoV-2 to attach, and then they get the SARS-CoV-2.
yeah. And the interesting thing about this paper is that this mouse strain, this virus has been around since for like I think 15 years or something since the first SARS-CoV outbreak because that also used the ACE2 receptor.
It's a reagent that they had before.
Yeah, so that's
Yeah.
so they already had this like adenovirus ready to go,
Hmm.
and yeah, so that's kind of interesting.
I mean, I think that that's just a great example of like how basic research builds on each other,
Yeah,
right? How the results from the past like build to the future, and why we should be asking all these different questions and like having that robust basic thing. Because like when you need to ask a certain one, the reagent might be around for you.
yeah. I mean, speaking of that, I mean there are like humanized mouse lines that do express ACE2 receptors, but I think
Hmm.
the reason that they haven't focused on that too much is apparently they're not very easy to get a hold of, and also like again we've got this inbred mice which are a great resource, but the issue is that sometimes that genetic like bottleneck can have like mean that the results aren't necessarily generalizable. So being able to test it in multiple mouse strains. So if you want to work on a mouse strain that has a deletion in a specific knockout in a certain gene or have some kind of change to the immune system, you have to go back to the drawing board and redo everything. Whereas
Well, you could do a
you can
cross.
do a cross. Yeah, sorry, you can.
Yeah, you could do a cross,
Yeah,
right? So you could cross in like so like that idea like um working with mice like when we were talking
mm,
earlier like mice are this huge genetic resource, right? Because like we have all these different inbred lines that
yeah,
have like a certain thing in them. And they get passaged they get inbred over many generations until right they reach some level of fixation,
yeah,
right? In that, as you said, they're almost clonal, almost
yeah,
identical. And I mean, you could then take two lines that have the traits that you want,
right,
right? Breed them together and continue that. And again, inbreed them for enough time that they come to some fixation with the two loci that you want. And then you'd have that cross line. But you can imagine throwing a virus in a pre-made mouse, right?
yeah,
Is a lot quicker than that process.
yeah.
Um,
So I think that's the main argument here is that this is just a lot quicker than doing that. And with coronavirus, time is of the essence. We're still in our living rooms and we want to get out.
Yeah, I think I mean the other reason that I read too, because the papers have been published where they infect the human with they infect the transgenic mouse with this, and again this is where the I read the stuff about the brain infection,
Yeah,
that like with that transgenic mouse they die from some strange brain infection, and again that could be maybe changed if they titrated down the virus a little bit, played with the model. But that's another rationale. They said let's just make another model as
yeah.
well to help deal with that maybe.
and that's what they do here. So so the interesting thing is that they actually they give you the first. So every paper has a first experiment that they never show you, the experiment where everything goes wrong, and they show us that in this paper in their first figure. They basically they say, okay, when we started out, we wanted to create this model, so we decided to look at all these immunocompromised mice strains, these mice strains that don't have a good immune system, because maybe since they don't have a good immune system, maybe they'd be like they'd get coronavirus. And what they find was that actually they don't. They seem to do quite well, even if they have the coronavirus. So
yeah, like they're all gaining weight normally, mostly.
Yeah, they're all gaining weight. Some of them are gaining loads of weight. They're real, they're really getting nice and plump mice. Yeah,
and they're not seeing that much viral replication.
Yeah, and some of these mice are mice that don't have T or B cells. So you'd be
the rag knockouts.
Yeah, the rag knockouts. So, and like when
those are very immunocompromised animals.
Yeah, you do not want to breathe near them. I remember working in the lab, you had to have like a full face mask, clean everything, and if coronavirus doesn't hit them, then there's something else going on, and so naturally I really appreciate the researchers like kind of publish this figure and showing their failures, because it's always.
But I think it's also important from that standpoint of establishing what is the model, like what is driving the issue with infectivity in this model, right? It's not anything about the immune system, right? It's about something else, and so they're gonna help us fix that.
Yes.
This also helps. Like, this data is also nice in light of the paper we just read, right? Because again, that same information still holds, right? That like in all these different mouse backgrounds, right? The infection's not taking hold, so it's something else.
Yeah. Exactly. And so their solution was to like use this adenovirus that holds the ACE2. So they did a test in a specific cell line that escaped me. I think it's Vero cells. Let me check. No, it's 293 cells, where they look at them and.
That's a lung epithelial.
Yeah, that's right.
TA2.
Yeah. So they got these to express ACE2 using their vector, and so you've got and FACS, which
so they feel like the vector works.
Yeah. And they got like it to express in the lung of mice. So this is like, so they use this term days relative to SARS-CoV-2 infection. So at this point they'd already decided to infect these mice, but this was like their test to see how much ACE expression they're having.
Yes.
So yeah, that looks like it's pretty good as well. And
yeah. So wait, so you're saying C is in cell culture? Or
C is in cell culture. So HEK293 cells were transduced with adenovirus HAC2 GFP, and flow cytometry looked at like HAC2 expression and GFP, and analyzed 4 and 10 hours after adenovirus transduction. So like
so that's cell culture. That's just making sure their reagent works in C, and then D they actually put it in the mouse and they make sure that the reagent works in mice.
Yeah, so the way the great thing about this is that you can actually see the timeline. So like the first thing that lights up is the adenovirus, and then after a while you see the ACE2 appearing. So 4 hours it goes towards the right, and then after a while it goes up. So all of them are so that's actually quite a good thing. So you need to make sure that your vector works in the first place.
Yes. And the dynamics of it, so that they can time when they're going to add in the SARS-CoV-2.
Exactly.
Because if they added the SARS-CoV-2 at the 4-hour mark, they might not get as many susceptible cells as they would at the 10-hour.
Yeah. So, and you can see that they've actually like decided to leave a few days between the two infections, which I think is a good idea. And then the next one, they look at the so this is looking at the inflammation caused by the adenovirus, I believe that. So
yeah.
So yeah,
they want to make sure that there's no adenovirus related pathology.
Yeah, and they also like do this strategy of using this IFNAR, like this antibody to knock down interferon, so to help the adenovirus along, so that it infects more cells. So IFNAR 1 alpha, I think it is, and they like show that they can infect more cells if they add this IFNAR. So and they stain So they
Yeah, I feel like at some level too, like A is supposed to help us understand this a little bit as a control, like
mm,
not a good one, just a rough one. In that, stat knockouts, like I don't think they're signaling through if not
yeah,
through interferon. So any effect of this interferon shouldn't be the thing that's helping the virus, helping the SARS-CoV-2 actually attach, right? It's
right.
still like it's the ACE2 that we're looking for.
Yeah, and I guess like they also want to make sure because the adenovirus it doesn't replicate, but they want to make sure that first time around it has the best chance of hitting as many cells, so
Hmm.
it doesn't get cleared out by the immune system too early. Um,
Hmm. Yeah, I just want to say, like, I think it's hilarious that we're talking about this paper, and this is the strategy they use because it just shows you how flexible these gene delivery platforms
yeah,
are. Adenovirus is also the platform for delivering the vaccine constructs into
yeah,
humans. Not this adenovirus, right? But yes, a replication deficient
yeah,
adenovirus.
exactly. So it's interesting how these things pop up, and it is
Hmm.
is really cool to be in it because essentially like this is like a form of a gene therapy that
Yeah. Yeah, we're giving the mice a gene therapy to make them more susceptible to a disease so
yeah,
that we can use them as a disease model.
yeah, I feel like someone in the 80s wrote a science fiction like horror story about us. I mean, it's
Yeah.
oh.
Oh, I mean, I had an old supervisor who said if God is a mouse, then we're just screwed.
Yeah, but on the other hand, we all see an immortal mouse far earlier than we see an immortal human. So
That's true.
so I guess it's swings and roundabouts, but
Oh yeah, do the ends justify the means? Okay, digression.
yeah. So yeah, I guess oh yeah, so we'll be moving on to figure two, um,
Yeah.
where they have various like strategies to try and they pick out very so 2A shows different strategies they're doing to create an infection. So there's
Hmm.
one where they add the IFNAR and the adenovirus as a control, and then they've got a couple where they add it. Like there's one time where they add it before the SARS-CoV-2 infection,
Hmm.
and they also like do intranasal and intravenously because I guess they're really desperate to get an infection. Um,
Hmm.
which again,
Yeah, the intravenous is really interesting because I think that people haven't said that there's a viral load in the blood.
hmm.
Um, so yeah, it's a strange one.
Yeah, think like this goes back to the idea of systemic infections causing a much more severe disease. So the idea is if we trigger that from
Right.
from the get go, we can get that really severe version of the SARS
Hmm.
coronavirus because that's at the
Hmm.
point where like your immune system's shutting down, like kind of triggering that like massive immune system action
Yeah,
early on.
the sepsis
Yeah.
element. Hmm.
Um, so yeah, and they do some where they like give the IFNAR before the adenovirus, sometimes when before the SARS-CoV-2, so to give the SARS-CoV-2 a better chance of causing an infection. Um,
Hmm.
yeah, and
They don't really see a lot of difference in the titers after that, right? Or in the PFU
yeah, not many. I mean,
through those different styles.
yeah, I mean, so next one they look at like weight changes. They do see some variation, but it seems like the one where they get the most adenovirus infected, but is the one that like seems to be the lowest. But I think there's not much like between that and the one. So I guess the point is I think this will be a bit more clear on the titers where yeah there's not much like different daylight between like all these.
Hmm. Yeah.
So
Yeah, like when they give the interferon, doesn't matter so
yeah,
much.
it doesn't like affect the SARS-CoV-2 infection so much. It might have a beneficial effect on this adenovirus one, but
Hmm.
yeah. Um, So that's, I guess, an interesting question for another day. D looks again. We got this whole organs. So like we looked at in the previous paper, where they're looking at
Hmm.
all the organs to see like if there's dissemination, and you get
Yep.
some things like the spleen. A lot of infections, and you see in the spleen because the spleen cleans up the blood, and there's a lot of immune
Hmm.
cells that kind of pick up viruses and stuff. So if something's in the
Hmm.
spleen, we generally think, oh, it's gone systemic
Yes.
because like sometimes you can like go through tons and tons of blood and never see a single like virus cell because there's just so much there, or it's weird. Um, but spleen
Uh-huh.
collects up quite a lot, so it kind of concentrates things. Lung, we've got a lot. Heart, again, we've seen some in the heart. And also some in the
Yeah, that
brain.
heart keeps coming up.
Yeah, that's interesting. We're seeing some in the brain.
Which again, that's like right because they knew because of the humanized mouse model. So
Yeah.
like it's something. I mean, again, you even hypothesized in the last one it could be because of the olfactory neurons.
Yeah. I mean, okay, you're right.
Thanks.
So, sorry about that.
No worries.
Yeah. So anyway, yeah, they look at intranasal versus IV, and they find that there's not much difference between them, which isn't. So they give like low dose and high dose, and they seem to be in the same like region. And yeah, yeah. So intranasal might be slightly lower, but I don't think it's enough to say that it's.
Yeah.
And I think
I mean,
realistically,
this is
we
great.
want.
Like they're really going in depth, right? Like they have these mice that they can work with, and so they're going to show us, right? Like all the things that could contribute to the disease. And argue for why they're going down one path versus all these others.
Yeah, I get the impression that this was like almost all these experiments were done simultaneously. They didn't know which one would turn out. And
exactly.
The intranase, it's like in the chimpanzee studies where
yes, where they just did all the routes in order to get disease. Similar, I think, is what's happening here. I agree.
Yeah. So like exactly. Yeah, and so if we move on to lung titers in Balbc and in CVF, so two different mouse strains they look at here.
Mm-hmm.
Which so Balbc is they're both inbred strains, but the difference in that like Balbc are white mice and black sixes are like black mice, I guess.
Yeah. Well, and it plays out in like other loci that are fixed in them.
Yeah, there's lots of differences between them, but the idea here is that you can use different mouse strains, and then you can basically get the similar disease. So that's actually, for any model, you want it to be reliable. And these papers really this figure looks really into the reliability of everything.
Yeah. And I think also it helps from that perspective of they're developing this tool to like recreate the susceptible mouse in any mouse background, right? So that you can go and study it. So this is helpful from that perspective as well.
Yeah. And H is lung titers, but after a long period of time. So after 8 days, 10 days post-infection. So I think as we remember, like the big peak was at day 3, and now, and this shows that it's still hanging around, which again is quite an interesting. Well, it's quite good because that's almost like what we see in humans, where like there is some small virus hanging around a bit.
Right. That's quite a bit. 5 to the 10.
Yeah, that is quite a bit. And they're titers as well, so these are live like viruses that are still hanging around.
Yeah. So it's like it really is hanging around quite a bit.
Yeah.
And that's H. And that is the SARS-CoV-2, or is that because it could also be titers for viral.
Yeah.
Oh, I was just wondering. It could have been adenovirus, but it's not. It's yeah.
Yeah. And then on I is we've got another big histology picture, which again like it shows like SARS-CoV-2 causing an infection in the lungs, and you can sort of and it's got like two controls: one with the adenovirus only, one with uninfected, and again what we see is that like the SARS-CoV-2 is causing like
the immune infiltration.
Yeah.
I like this one because it's good to know that the adenovirus only doesn't cause any immune infiltrate. I don't think they really. I guess they showed us that the mice are fairly healthy from like the gross standpoint, but it's good to know at this sort of molecular resolution, histological resolution, it's also the case.
Yeah, I think in the earlier figure where they're looking at the adenovirus, there was some small like kind of infiltrate, but some small like kind of immune response, but it wasn't that big. Nothing like what we see here.
Yeah.
So that's quite important. This is and in figure, this one is figure.
Yeah, I mean, in comparison, right? Like between in this eye, yeah, I see what you're saying. Like in figure 1E, right? There is some things happening, right? There's what are they staining with here? 1E. Um, SARS H2 positive cells. No, they're not. They're not looking at. They're looking at just the ACE2 positive cells in that one.
Yes.
Yeah.
Yeah.
Yeah. So this would be the first time that we see, because now they're not looking at ACE2 positive. The brown is
The SARS, I believe. Check this quickly. They
figure 2I.
figure. Yeah. So, yeah, figure 2I. Yeah. So that's yeah. So yeah.
So
So
what's the brown in this case?
the brown is hold up, because this is weird because like I think some of the like figure legends are on multiple pages.
Yeah,
So
I know. I'm just like
it's a
scrolling through. I was
bit
like, it's not super easy to,
yeah.
but I don't see it in
SARS-CoV-2
this one.
SARS-CoV-2 RNA is what they're standing for. So it's in
Ah,
situ hybridization. So they
right.
are looking at the SARS-CoV-2 DNA.
So we're still not answering that question
Sorry, not
necessarily.
DNA, RNA. Sorry.
RNA, yeah.
Yeah.
Yeah, so this is like obviously they would see this because they're giving all of it. I guess they're seeing replication, which is
Yeah.
nice. Yeah.
Yeah, because I think like because again we are missing that control of the SARS without the
Mm-hmm.
adenovirus treatment. I think they might deal with that later on in the paper, but yeah. We're going to have quite a bit more like histology to look at. So because in Figure 3 it looks
Oh yeah.
so yeah, Figure 3 looks at SARS-CoV-2 infection, looking at biopsy mice after no treatment or after intravasal administration of ACE2.
Mm-hmm.
So they've got like, and B looks in at like
So
yeah,
this is what I was thinking that one was. That one's just
yes,
does the virus get in, but here we're looking at the actual pathology of the immune infiltrate.
yeah, and like what we're seeing in Figure 3 is it's seeing like it produces some and they try and they basically make the argument that like kind of after a certain period of time the immune infiltration like goes down after apart from, but in some cases like in Figure 1C like where they've add the adenovirus but with that IFNAR at the same time and the
Mm-hmm.
SARS-CoV-2 that causes a real big like amount of infiltration.
Mm.
So essentially the screen turns purple with the amount of infiltration.
Yeah, I mean,
Um,
it's definitely more 8 days post-infection. Like, the bottom, the lowest corner. Oh yeah,
yeah,
what you're
the
showing.
lowest corner is like you can so each of these are like more zoomed in versions of each one, and the lowest corner you can see like vessels are like being clogged up with all these infiltrates. So this would
Hmm.
be not a very happy mouse at this point, I wouldn't think.
Yeah. Yeah, but I would say, right, see, the thing is that even 4 days post-infection B, right, figure 3B, right, like there's some foci, I would say, of immune infiltrate just from the
Yeah,
adenovirus. So, like, the
yeah,
adenovirus is causing something, and then as the infection continues, I guess we're presuming that is, yeah, it doesn't get worse, right, in the 8 days post-infection. But yeah,
yeah,
with the people that get, or for the people, the mouse that get the SARS-CoV-2, it's definitely getting worse.
yeah. Uh,
Yeah, it's more
so
widespread.
Yeah, I think that's like an important comparison, and I think that'd be the question that everyone would have. Like, what's the adenovirus doing? I mean,
Hmm.
it's not supposed to replicate, so it shouldn't be too severe of an infection.
Right.
But we want to dot that I and cross that T.
Yeah,
Uh,
absolutely.
yeah, and so I guess this is now that we've established the model. Next step is to see what it can do.
Yes.
Uh, so instead of testing out a vaccine, they test out an antibody treatment. So
Hmm.
so the
Yeah, so that's an antibody made outside of the body, right? Like biotech style, and then given as a
yeah.
drug. There's lots of these drugs now, right? Biologics is what they get called.
Yeah,
And you can have them for infectious disease as well. Although I don't think there's many examples of that.
I mean, I think like there is like I can't remember. There's one that appeared on House quite a lot. IVIG, I think was where like intravenous immunoglobulin.
Yeah, because
So
that's for broad broadly immunocompromised people. You just get all the
Yeah,
IGs of somebody. But in this style, they're using a monoclonal, so it's like specific
yeah,
for something. Um,
yeah.
mhm.
I mean, we do have like monoclonal like treatments for certain diseases like Herceptin or other things like
Right.
that. Or
Right. But in
I
those
mean,
cases, they're not infectious, right? Like the
yeah,
targets of them are not infectious targets.
yeah. Um, Yeah, and in this case, 1B07 is something that targets the receptor binding domain of the spike protein,
Hmm.
and they use an isotype control that it hits another part of the spike protein that doesn't neutralize the virus. So
It's nice
I think
control.
it's a nice control. It's nice
Really
that
nice
they have
control.
that control.
Yeah.
Uh, yeah, and again, they test the neutralization in zone B. They test neutralization in some like cells. Let me see what noted down what cells they were. I did not note down what cells they were. I need to read the paper.
Vero
Vero cells.
cells.
Ah, good old Vero cells. Green monkey kit cells.
Yep.
Um, yeah, and they also looked at
It
like weight
works.
loss.
So it
Yep,
works.
it
It works in
works.
cell culture.
Yep,
Um,
those are very nice curves. I like those curves. Weight change. It looks like the mice that have that antibody, they're doing well.
mhm.
They're gaining weight.
So this is
Um,
this is interesting in comparison to the last paper, right? Is that in
yeah,
this disease, the mice are losing weight.
yeah.
Mhm. And
Uh,
that's probably because, yeah, I guess they're getting a more severe infection.
well, yeah. I mean, I've got an idea about that because because with the natural mouse, they're only expressing ACE2 on the cells that would naturally express ACE2,
Yes.
whereas
The tropism here is like off the rockers, maybe, right? It could be all
yeah,
sorts of
that
strange cells that wouldn't normally get infected.
exactly because
Mhm.
they put this adenovirus under a site under a virus promoter, so that means that it's expressed in whatever cell it hits. So
Mhm.
that means that
Mhm.
though all so this goes back to that first paper where we had that lovely like picture of like the SARS-CoV-2 going specifically towards like the club cells, and
Yes.
but we
Yes.
wouldn't necessarily get that with this one because the SARS-CoV-2 would be attached to anything that was expressing
Right.
the protein. So
Right. And it could be like sort of whatever cells are most susceptible to the adenovirus infection ends up being the cell type that's going to be infected by the subsequent SARS-CoV-2 infection.
exactly. So actually, this could actually be a somewhat different disease to what you'd expect in mice,
Hmm.
and that could explain why it's more severe because more cells it will mostly be cells in the lungs, but it might be different cells to what would be
Sure.
ordinarily be expressed.
Sure.
But
And again, it comes back to that endpoint. Like if all we're concerned about is seeing that immune infiltrate and saying like that is our correlative disease, right? Then we got it here. But if we are looking for something more subtle, then maybe this is not the best model because we're just
yeah,
blasting every surface that comes in contact with the adenovirus with susceptibility.
yeah, I mean, yeah, I guess it depends on what you're looking to get out of that model.
Hmm.
So if we want to study the underlying pathogenesis of like kind of what is the SARS-CoV-2 actually doing inside the lung cells, the other model might be better because the lung cells are behaving in a way that's normal, whereas this one might be treatment like oriented stuff. Like if you're looking at late infection stuff that's like where things are getting very severe, then this might be quite useful in that sort of aspect.
Or
Um,
like again, like they're envisioning some sort of like I guess immune study, learning how like which immune systems are important, right, during the infection. um
yeah,
but again, everything comes with its limitations. I guess is the important thing to remember here, right? And so like this is a specific limitation of this model, and the limitation of the other one, it's still there because it's not exactly human epithelial, right? Like
yeah,
style expression, but it's a lot closer, and they showed us evidence of that. Here, we
yeah,
don't see the evidence. Yeah.
yeah. Uh, and so we have lung titers as well. Again, we're
Hmm.
seeing like titers and loads, and again the 1BL7 treated mice seem to be doing a lot better in
Hmm.
terms of like, and we're also seeing that kind of difference between the amount of active RNA and the amount of active uh, like viruses, because again that doesn't necessarily correlate.
Yes.
Uh, and I think that's and we've got figure something. I don't know, we've got like some more histology to coming up as well. So the final histology is looking at
Oh wait,
Uh,
did we say that you did the viral RNAs, and then we also have all of these cytokines panel that they did.
yeah,
Uh,
that's true. So yeah, because they do talk about like the 1BL7 versus the isotype control. And it's weird because they split out like it into Figure 5a, but in the paper it comes in quite. They talk about it a bit earlier.
Gotcha. We're just
And
gonna jump to the.
we're just gonna jump ahead to one where they look at 4 days post inoculation, and they find that okay, yeah, more SARS-CoV-2 RNA in the isotype versus the 1BL7.
Mm-hmm. And
Um,
again, also looking at the histology here. I mean, they don't
yeah,
at this time they don't have the independent score. It'd be nice to have the independent scorer here,
yeah,
right, to show us those little bar graphs with these. Mm-hmm.
yeah. Because we only see like one panel. I mean, off panel it
Mm-hmm.
could look completely different. So
Yeah.
we kind of have to trust that these are representative.
Mm-hmm.
And but again, same with the graphs. So there is always a bit of trust there. And
Yeah.
yeah, and yeah, and so okay, right. So also moving on back to Figure 4, to the Figure 4F and G. So these are all also looking into dissemination. So are they in
Mm-hmm.
the heart or in the spleen? And this one, it seems that there is a bit more because again they're looking at viral loads, so this isn't necessarily representative of real live viruses.
Mm-hmm.
But yeah, it seems that there might be something there. There might be some difference there at least. So that's a positive thing. And if it's a antibody therapy, the main difference you'd see would hopefully be in the serum or in.
Mm-hmm. Yeah, it's interesting that the difference doesn't seem as pronounced as in the lung.
Yeah, I mean,
Mm-hmm.
I mean, part of it could be because there's much less virus out there, so the differences are there's going to be more variation. Because when you have a small like kind of, yeah, I don't know
Oh
what I'm
yeah,
saying
it's
there.
true. Yeah, yeah. Well, I
Yeah,
mean, like the spleen clusters really tightly, but the heart's all over the place,
yeah, aren't all our hearts
right?
all over the place?
But I guess that's probably also to the fact that this is not like the preferred area for the virus, right? Like
Yeah,
there's some other conditions that we're not really controlling for understanding that's causing this dissemination.
yeah, and I think like ACE2 is expressed in heart cell in other like kind of cell lines, and we don't
Mm-hmm.
necessarily and we're not sure whether this virus would have hit those cells,
Mm-hmm.
and so how this virus behaves systemically outside of the lung might be somewhat different to what it would behave in humans,
Yeah.
and then there's
Sorry,
H, which
I totally forgot. This is all with intra intranasal. Yeah, this is all
yeah,
intranasal.
yeah, all in intranasal. So
So it could also be like um, technique. I mean,
yeah,
there could also be some technique thing going into here where it's like it was just given in a different way, maybe.
oh yeah,
Um,
intranasal like the
tubes.
actual volume does make a big difference when you give it to mice, because if you like give like say only like say 10 microliters, it'll be in the nose, but if you give up to like 40 microliters, it'll definitely hit the lung, and 50
Yeah,
you're almost drowning the mouse, so
right,
don't try not to do that.
right.
Yeah, so there is a lot of technique into that, because again sometimes you accidentally, if someone's like giving it, they can put it straight into the mouth's mouth, and then suddenly you've got a slightly different
Gas trail.
model.
Yeah, yeah.
Yeah, so I mean this, because I know like when I was infecting mice, sometimes they would like really lap up the virus if they were like still awake, and I'm like, no,
Not for drinking.
not for drinking. But yeah, and yeah.
uh, oh,
Oh,
moving to
sorry.
H. Or you want to say something else about?
Oh no, I've basically said everything I did. But if you want to say something more, then I can jump back.
Oh no, no, we're good.
Okay, cool. Because Figure H is another one of those big like cytokine panels.
Hmm.
Like in
Inflammatory cytokines go up, but ask us on the details of what that means in all these different cases. We're a bit more fuzzy on
yeah,
those details.
immunology is confusing. Different like because it is probably one of those. I mean, I like to think of myself as someone who's like very. I like immunology, but I'm not good at it. I mean, I took immunology like kind of subclasses for my entire BSc. I was like every time I had a chance to do anything specifically immunology, I'd definitely do it. All the classes that I did very badly in were all immunology ones. I'm passionate about it, but I'm not good at it.
Yeah. I mean, it's tricky, right? Pathogenesis is like very tricky to study in that sense. You have all the side of the pathogen to study, and then all the side of the host, and disease is somewhere in the middle there.
Yeah, I mean that's why it's so interesting because like I mean, to me at least it's a mystery. To an expert it's normal, but for me it's like, oh my god, this is like all these different connections, all these different networks are
Yeah,
happening, and we're just getting
yeah.
a small snapshot from these cytokines. And I'm saying, oh, it's generically like inflammatory, but obviously there's a lot more detail you can dig into with each one. I
Right.
mean, each one of these could be their own paper if you digged in deep enough.
Yeah, absolutely. And characterize the pathway and the mechanism in which that upregulation would affect the infection one way or the other.
Yeah, exactly. Because everything your immune system does is a series of decisions made by different individual cells, which can lead to like an outcome, which makes it
Yeah. And what we're seeing here is just a snapshot of everything that's being said, not by whom or to whom in the
exactly.
immune system, just like out in the ether, all of those chemical words, oh, they're being said.
Yeah. I mean, at least when you're looking at neurons, you know where it's connecting and who's communicating with who. With immune cells, there's like all these chemical signals that you don't know
Right.
where they're going
Well,
to. So,
not to say that you can't know, though, right? Like
oh
there
yeah,
are methods of knowing. Like there's ways of fixing the cells and looking for the co-stains, right? Like
yeah,
cell and looking at doublets or something like that, or doing in situ staining, right? And
yeah.
co-localizing the signals that way. But
I
yeah,
mean,
these graphs
yeah.
aren't that method.
Yeah, I mean things like that's why fax is such an amazingly powerful tool, and
Hmm.
why, yeah, and these sorts of things are interesting. But I think they paint a broader picture of the disease that's caused in this instance.
Hmm.
Uh,
Yeah, but by giving this monoclonal, they do see suppression in also those immune responses, right?
yeah,
So less disease is happening, right?
yeah,
Less virus is being made and less disease is happening, less inflammation is happening.
yeah. Which I think brings us to the final like pick of the of Figure 5D, where they look at of after 8 days, where they see that what was my
Five
point?
5B.
5B, yes.
Hmm.
Um, where uh, yeah, they look at it staying long. So yeah, looking at kind of the recovery of the mice essentially. So because I think this is after the worst part of the infection, and they see that the because I mean your lung gets destroyed during an infection, but it does repair itself,
Hmm.
and so we see 1B07, they're kind of returning to what we think of as normal and healthy lungs, whereas there's still some kind of inflammation going on.
Yeah, I would say there's a lot more infiltrate in the isotype.
Yeah,
But like the structure still seems pretty wrecked up to me.
yeah,
Again, this would be a great time to show us the bar graphs and have some histologists weigh in on this. Um,
yeah.
but
I
yeah.
mean, yeah, I mean, the thing that always shows up for me is whenever you see the ciliated cells, because when ciliated cells get inflamed, all the cilia like kind of disappear or they look really ragged, and then they regrow
Mm.
afterwards and look really nice. I think you can see that somewhat here, but I'm not. But mostly around the like the cell lining is where things get where a lot of damage happens, and yeah, and here you can sort of see the inflammation around those like kind of like so
Hmm. Yeah.
uh, and of course, like in this case, the information can be isn't just like them fighting disease, but also can be repair and rebuilding the cells and like cells
For sure.
regrowing as well.
Hmm. Hmm.
So
Yeah. So like in the later time point, right? Like inflammation is just whatever, right? And in some ways, the disease that we're looking for is inflammation at a certain acute time point,
hmm,
because that's what is correlating to the ARDS.
yeah,
Um, yeah, but yeah. So that's this paper. Do you have anything else to add about it?
I mean, yeah, I think this is we've covered most of the things, and I guess the thing that we'd add is like the compare and contrast, like the final because we've got
Hmm.
these two papers. What do you think?
I mean, they both like it's very. I think what I'd like people to take away from this is seeing that they're essentially they're using the same spike and receptor interaction, right? That is the thing they have to change in order to get the disease to happen. They're first of all they're getting at that in two different ways. So that's like the the sort of the synthetic biology side or like the bioengineering side of it, right? Different ways to get the same model. And the readouts they use are also a little bit different, right?
Yeah,
Although they're still again getting at this idea of inflammation and in the ways in which they've specifically shown the type of inflammation that occurs in these models and the type of disease that occurs, it tells us something different about the ways that those models might be used to tell us about human disease, right? And the limitations of each model on human disease itself.
Yeah, no, I think that you really hit on all the points that I was about to say. So,
Oh.
no, that's
Yeah.
fine. That's great. It saves me saving time. But yeah,
they do the panels are the same between the two,
exactly.
right? The histology is the same. So much is the same. So like it's like yeah, focusing on these small differences that we're seeing, right?
Yeah.
The time and then the application, right? That the
Yeah.
virus can be applied in very different circumstances versus the adapted. Yeah,
Okay.
I mean, it's just going to be up to where we see the application papers like end up
Yeah.
making use of these.
So let's do like a thought experiment. So let's say you're
Hmm.
in the lab and you had a choice of which of these models to use to say study pathogenesis.
Oh
What would you
yeah.
do? Yeah.
Well, I guess like everyone's limited by like costs and things.
Yeah.
Study pathogenesis, meaning I want to know how the virus is causing disease, or am I
Well, I
studying an
guess
intervention?
yeah, that's a difficult one because I know that if I was studying disease, I'd plump for the first one because
Yeah.
that gives you the right tissue tropisms. But if I was studying like a treatment, then I might actually try out the second one.
Yeah. So like for yeah, I would definitely say that because I when I was studying like staph disease, right? Like
Mm.
I was obsessed about the earliest moments of that
Yeah,
disease, right? Like what gets set up at the beginning that then causes something to go out of control. And so for me, yeah, I like that idea that we could see the types of cells that are infected inside of the mouse adapted model, and then potentially you could like sort out the cells, the specific cells
hmm.
that were infected, right? And look at some of the signals that are coming out from that original sort. And if those are similar to, I would love to pair that with
Yeah,
like the autopsy, like ex vivo, right? Like autopsy people that also have a similar response to the virus in their sorted cells. I would like that. So I would use the mouse adapted one if I was just given like carte blanche. But I guess it really does depend, right?
yeah,
Because that's not everyone's question. Like that's my
exactly,
question, and I'm not being asked to defend the funding of it or like or,
yeah,
like all this sort of stuff. All I'm going after is like what I think where I think the most important steps of disease are, and I drill down to this. But whether or
yeah.
not that's the best way to approach it, given the constraints of all the materials and things, that I don't know.
Yeah, I mean, because during my PhD, I was very interested in dissemination, in like the systemic aspects of the disease.
Mm.
But in that one, I feel like I would go with the first one again because with the tissue tropisms and all the ACE2 that are expressed in other cells, I still
Mhm.
want to have that relation still happening.
Yeah,
So
you wouldn't want, yeah, even in the disseminated form, you don't want to stray too far from like just blasting the phenotype of the disease, right? Like
yeah,
you don't really care for that immune infiltrate per se. You just want to know what else is happening, could be happening in the body, right? That relates to human disease.
yeah. I mean, I feel like the second one. I mean, it's great from a perspective that we are at the stage where you can use like gene therapy to change all the kind of genetic makeup of mice. I think that that's really important, really interesting. At the
Mhm.
moment I'm having trouble finding out situations where I'd use that over the other one, but uh, I know that there are situations that would exist.
Well,
I'm
what could be interesting is that, as I said at the beginning, I thought that when we were choosing the mouse adapted model, we were choosing something that was like published after the preprint. But there's a
mm,
preprint, right? That was another mouse adapted
yeah,
SARS, and in that one, it's in bioarchive, Dinon et al. I put it in
yeah,
the background folder, but in that one, I don't think they're not passaging to do the adaptation. They're using like a different version. They're editing the virus
yeah.
so that it just has a different receptor binding domain.
Yeah. Oh yeah, I've just thought of something because again, if you just want to look at the different strains of SARS-CoV-2 caught in the wild, you can't use the mouse-adapted strain. So if you're asking a question of whether one strain of SARS-CoV-2 is more infectious than another, then actually the
Oh
ACE2
yeah,
model, yeah,
then you'd have to go to the ACE2 model.
yeah,
Yeah.
because you
But
can't,
what's
yeah,
nice, I hope, is that I mean, as far as I know, at least there are no variants of biological relevance, right, that have emerged for SARS-CoV-2. But
yeah,
yeah, in the case that there are two candidates, and we know that like there could be things that drive it, right? If we see
yeah,
mutations in that receptor binding domain, right, or even in even in accessory proteins could drive tropism,
Yeah.
it would be, yeah, this would be the model you'd have to use instead. Yeah.
Yeah. So
Yeah,
I get-
that's a really good point.
yeah. So I guess like the thing is like these mass models are the basis, and you're allowed to have a much more imagination when you're working. It's more basic science, so we can actually delve into the basic things with them. So that's
Yeah,
why.
I mean, these are the tools, right? Like
Yeah.
these are the tools, and how you put them together. That's the puzzle. That's the experiment. And so in these papers, we're presented with tools, and they've tried to characterize them for us by showing us how that disease works, by showing how they did it. But then how other researchers will take this and use it in their lab, that's up to them, right? And that's kind of the awesome thing about, science and like making research tools, is that they get taken up by other creative minds to just figure
Yeah.
out answers to questions.
Yeah. I think I've basically said all I can on this at this point.
Yeah.
How are you feeling? Yeah.
Yeah, no, I'm pretty happy with it. So yeah, this is Micro Twitter Journal Club.
Yep.
This is our on weeks where we dive into the papers and we look at the figures. But on our off weeks, we look at the news and look for articles to read. We track all of that in our Zotero, which is a reference manager.
Yep. That's in the
And
description.
you
Yep.
can communicate to us through Twitter, either by our tags or the hashtag.
Yeah. Thank you so much for joining me. Hopefully we'll see you next week. Where on our off week we'll be looking at the first looks at papers and microbiology news.
All right. See you guys later.
See you later.